Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about how you can design your own primer for your PCR reaction, and by doing so you can perform your PCR and followed by restriction digestion of a particular gene. So let's jump into the topic. So at first you have to open your browser and type here SNP, that is the database for a single nucleotide polymorphism. So whenever you click the first link that brings you to the SNP database, that is for NCBI database. So here just type your particular gene of interest or particular SNP of interest. So here I take one example of one SNP, that is RS2285053. So whenever I click that particular SNP, so it appears a new page and it shows that particular SNP ID and also this name of this particular ID and all of the details of this particular SNP. Here you can observe the particular RS number, that is the SNP, the single nucleotide polymorphism ID. In here you observe that is this particular SNP is C substitution T, and whenever you click on the show flanks, it particularly gives you particular flank of this single nucleotide polymorphism, and this is particular single nucleotide polymorphism of a gene that is known as MMP2, that is matrix metalloproteinase 2. So whenever you click the var view, so after clicking the var view, that is the variable view, it redirects into a new page and it takes some time to load. So whenever this particular page comes up, then you observe here this is the particular RS number, that is the SNP ID, and here from here you click on the tools and then go to the sequence text view. So whenever you click on the sequence text view, just enlarge it and you will observe that particular SNP, that is the single nucleotide substitution, that is the C substitution T. So in wild type it always C, that is RS2285053. So this is particular the nucleotide sequence. So now we design a primer for detecting this particular polymorphism in PCR RFLP reaction, that is a polymerase chain reaction followed by restriction digestion. So if you observe here in the left hand side, you can observe that there are several numbers indicates these particular nucleotides. So each of the line, that means each of the line consists of hundreds of nucleotides. Okay, so 100 of the nucleotides have each of the lines have 100 of the nucleotides, and you have to select. This is the thumb rule. Basically you have to select 250 nucleotides up and 250 nucleotides down from this particular nucleotide polymorphism. So here I select from here to at least here. Okay, then I simply just copy it by clicking Ctrl+C, and now I have to minimize this particular browser and go to my Word. Okay, so MS Word. So just open your MS Word and just paste the sequence. Okay, and whenever you paste the sequence, it appears like here. Don't worry, just adjust the sequences. Okay, now adjust the sequence, and this is the nucleotide sequence but not the FASTA sequence, but you have to create this sequence into a FASTA format. So how can you create this particular format into a FASTA format? Just enter and create these symbols, type the name of your gene. So here I type this particular gene name. So now this total sequence, so top to bottom, this sequence is known as the FASTA sequence. Okay, so you have to search or identify your single nucleotide polymorphism from here. After that you can proceed, that means from which length you can obtain your primers to get the perfect result of your gene of interest. So just go to the variable view and search this particular. Here you observe there is several nucleotides upstream of this particular single nucleotide and downstream of the single nucleotide. So you have to search like TGC GGAC. Okay, so let's copy these 5 or 6 nucleotides, go to your Word, enter Control F this and search it here. So whenever you search it here, that is TGC GGA, TGC GGA, and after that you again go to this particular sequence view, and here you can observe there is a triple C, that is C, C and C, then TCCT, TCCT. Okay, so CCC, TCCT. So here one is selected particular for your find, and here is CCC and TCCT. So this this nucleotide is your particular polymorphism. Okay, so mark it just here, and now we jump into the primer design. So basically open your browser again, go to new tab, and here is very useful website is there, that is the Europin Genomics website. So this is the primer designing tool, that is PCR primer designing tool from Europin India, so or Europin Genomics. So here just paste your FASTA sequence, just copy it from here and paste your FASTA sequence here. Okay, so after you copy your FASTA sequence like this one, next you have to enter a name. So here I also type a name, MMP2, that is 735. Okay, so I have put name on this, and here you can see that length is 869 bases. Okay, just open your Word, go to your FASTA sequence, and we only want 300 to 550 base pair long gene sequence, that is amplicon sequence, not much than that. So go to your primer designing tool, that is Europin Genomics, and design parameters, open design parameters. Target sequence you can set here from start to end. Whenever you enter the whole genome sequence or whole gene sequence in target sequence in FASTA format, then you have to type start or end, otherwise do not touch it. Okay, next primer criterias. Primer criterias are always same, that is the length is 18 to 22 nucleotides, GC content is 40 to 55, and melting temperature from 50 degree to 65 degree centigrades. Next, amplicon criteria. So here you can modify your need. That means I am here modify my need from 250 base pair or 280 base pair to 550 base pair. Okay, now I select my amplicon size here, and GC content is 20 to 80%, melting temperature is 70 to 95, it's okay. And then reaction conditions, if you want to modify these reaction conditions, then you can modify it, otherwise let it be. Output parameters are as usual. Then design primers. Whenever you click the design primers, it will take some time to create a primer for your particular amplicon, and when this process ends, then Europin suggests some of the primers that is essential for you, or that is you can use for your PCR reactions. After completion of the total process, you will get this kind of results from Europin Genomics, that is the primer designing tool. And here you can observe it gives us 5 different options or 5 different primer sets for our PCR reactions. So length of the 5 amplicon sizes are 412 base pair, 411 base pair, 407 base pair, 446 base pair, and 393 base pair. So you have to find out which template or which primer sequence sets can actually suits you best. Okay, so just take this sequence from here. Okay, now copy it and paste it to your Word file just like that. So it is our forward sequence. Okay, so it is our forward sequence, right? So you have to find out this sequence out of this total FASTA sequence. So just take 5 or 6 nucleotides, copy it and search it. Whenever you search, you will get 2 different positions, but you have to match all the sequences. Okay, so AG T GAG, AGT GAG always there, but here CG AG, so CG CG AG, then T 4A T 4A. So this is your sequence. Okay, so the length of this forward primer is 20 base pair, 20 base. So select it from like AG, this one, this much. So this much sequence is 20 base. So highlight it. So this is your forward primer sequence. Okay, now you have to find your reverse primer sequence, but means where it ends, actually ends. Okay, so where your reverse primer ends. So go to your primer designing tool, copy the reverse primer and paste the reverse primer, but you cannot find this reverse primer in this sequence or FASTA sequence. So what you have to do, you have to create its opposite nucleotides. Okay, so for T it's A, for G it's C, for G it's C. Now T T T, then G, and so on. So after you complete your reverse synthesis, that means you have to add your complementary base, because this reverse primer binds with the complementary strand that is not shown in here. So you have to select this complementary strand, and it is now not been able to detect from this FASTA sequence. So you have to tweak the sequence, that means you have to reverse it. Okay, so you have to reverse this sequence. So how can you reverse this sequence? Just paste one after one. So A is first here, adenine is, then C, then C, then T T T, then, so you have to complete this by reverse manner. Okay, G, then T. So after completion the reversing of this particular sequence, now you get this sequence, that is the original sequence of this FASTA sequence. So you have to find this sequence in the FASTA sequence. So copy it and just paste that particular sequence. So you get these 2 sequences right there. So you have to match the results. So TTT CCA, TTT CCA, then CTG, then CTG, then TCT, TCT, then, so this one is your particular sequence. So after reversing this particular sequence, that is the complementary to these reverse primers. So this one is the reverse primer. Okay, so after this one is the reverse primer. So you have to convert it into their complementary sequence. So this one is the complementary sequence, right? So I have to write it down, complementary sequence. Okay, so this is the complementary sequence, but you cannot find this complementary sequence in this FASTA sequence, because this complementary sequence is not present, because it is in the other strand. Okay, so you have to reverse the sequence, reverse the complementary sequence from here to here. That means A is here, C is here, then this C is this C here, then this T is here. So you have to reverse it by all this manner. Okay, so you have to reverse this total sequence to this one, 180-degree rotation. After that, you have to find this sequence TTTCCA. So copy it and just search it. So whenever you have to copy it and search it, then you find this one sequence. So here is your target sequence, that is reverse primer sequence. Okay, so this one is the 20 base, I think 20. Okay, so this one is your reverse sequence. Okay, so now we get our forward primer as well as reverse primer. So from forward here to here, last, the total nucleotide number is 393. Okay, so here you can observe here that there is a 393 amplicon length. So this particular segment we have to amplified by our PCR primer. So this PCR primer helps you to amplify this length of this particular segment. Okay, now you have to check the strength of this primer. Okay, so how can you check the strength of this primer? So again, go to your browser and search here NCBI. Whenever you search the NCBI, it opens the National Center for Biotechnology Information, NCBI database. Go to NCBI database and go to blast. After clicking the blast, there is a page appears, and here you have to click nucleotide blast, that is nucleotide to nucleotide blast. So whenever click the nucleotide blast, then this page appears. So you have to enter your FASTA sequence. So FASTA sequence, what FASTA sequence? Our primer sequence. Okay, that is the forward and reverse primer. So make this forward and reverse sequence into a FASTA sequence. So MMP2, that is forward, hit enter, copy your forward primer from here and paste it here. Okay, now again MMP2, then reverse, and now copy the reverse primer, and paste it. Okay, so whenever you paste this sequence, this is the FASTA sequence of your forward and reverse primer. Now you have to choose your search set or database. So we here we set human REPS-Q sequence. We select human REPS-Q sequence, gene sequence. That means we have to search about human genome. Okay, then click on the blast. Whenever you click on the blast, it will take some time and gives you a particular result of your selected primer. Okay, so here comes the result, and as you can see here, that is the Homo sapiens matrix metalloproteinase 2, that is the MMP2 REPS-Q gene sequence on chromosome number 16, and this is the e-value of this particular primer. So first one is the primer sequence, that is we put here in this blast, and this blast generate this kind of results, and you have to check this particular e-value. Whenever this e-value is lowest, that means your primer generates good result. Otherwise, whenever your primer have the higher e-value, then you have to get, or you will get various non-specific bands in your PCR reactions. Okay, so after selecting this your primer, you can also check from this method other primers also, like this one also. You can find all your primers, you can blast all your primers in the NCBI database and you can get your results. So this is the basics of primer designing. Okay, so now we have to jump in next topic, that is the restriction digestion of this particular gene, that is the particular SNP to RS2285053. So we have to go to our FASTA sequence. So here you can observe this is our particular SNP, that SNP is mutated. Okay, so the mutation is here, that is the MMP2 735C substitution T mutation. So whenever C is a wild type allele, okay, So C allele is wild type allele and T allele is mutated allele. So whenever this sequence here, this sequence, I have to copy this and paste it here. So I have to paste it here. So this is the single nucleotide, right, that is modified into this kind of sequence. So it is modified like this one. So T is modified by T, so C is modified by T. Whenever C is modified by T, here you can observe here is a particular sequence, that is GACTC, okay, CTC. So GACTCT, this kind of sequences appears. So you have to find which restriction digestion will digest this product. So whenever I search here, I have got my result. So actually HINF1 restriction digestion, restriction enzyme. So HINF1, you can search the sequence of this particular HINF1 from the database of NEB, so New England Biolabs. So particularly here you can observe here is the sequence GANTC, GANTC. N means any nucleotide, N means any nucleotide. So GANTC, this nucleotide sequence, whenever this nucleotide sequence is here, then HINF1 can digest this particular nucleotide sequence, okay, or this palindromic sequence. So GANTC, now go to our Word file. So GAN, N means here N stands for C, so this one like this GAN. So suppose we have to assume this is the N number of sequence. So GANTC. So here whenever the wild type gene is there, there is no mutation there, so HINF1 cannot digest this particular variant. But whenever GANTC, that means GACTC occurs, then HINF1 can digest and break this particular into this kind of thing, result, okay. So whenever HINF1 cut this sequence, HINF1 just cuts it from between G and A. So whenever G and A is cutted from HINF1 digests this portion, so you can get a fragment of like this one, okay. So 165 base pair, so 165 BP fragment, 165. Now then from here to here you will get the 228, that is 228. These two bands you will observe in your agarose gel whenever you digest this particular amplicon, okay, by HINF1. So it is digested by HINF1. So total amplicon size is 393, okay. You can check from this PCR primer designing tool, that is a 393 base. So 390 total base is uncut, so C is a uncut, so C is equals to uncut, uncut equals to the size is 393 BP, okay. So base pair, and whenever it digests, whenever it is mutated, that means the mutated allele is T. So T allele basically digest, so T allele, T is cut, T equals to cut, and T is cut equals to, it generates two fragments, one is 165 BP, another one is 228 BP, okay. 165 BP and 228 BP. So this is the total, and whenever there is CT polymorphism observed, then 393 plus 165 plus 228, those three bands will appear in your agarose gel. So in this manner you can design your PCR primer, you can check your PCR primer, as well as you can find your suitable restriction enzyme for your particular restriction digestion process. So hope you like that video. If this video is really helpful for you, please hit a like button, don't forget to subscribe, and thanks for watching.